All right, good. So, Anastasia, maybe starting with you, just introduce yourself, where you go to school, and then we'll have Dakota and Eleanor, followed by Dr. Gillette.
Can I interrupt a second?
Yeah, sure.
Introduce yourself. Um, Please let me know if you plan to go to college, and if so, maybe what you're thinking about as a major. This way I get to know who's there. If you don't know your major, that's fine.
Okay. Well, my name's Anastasia Worthy. I go to Cranbrook Kingswood, which is located in Bloomfield Hills, Michigan. I'm in the 12th grade currently, and I do plan on going to college at Wayne State University, and I want to major in pre-med.
Okay. Terrific.
Yeah. I don't know, whoever wants to go next.
Dakota, why don't you go next? So Dakota, I think, is going to write down. You're going to IM. So hopefully Dakota sees this. Okay.
Oh, there we go.
I don't know if Dr. Gillette can read this. I'll read it if you can't. It says, "My name is Dakota Willis, and I live in New Mexico. I go to Albuquerque Academy. I plan on going to college, but I'm not sure where. I really want to go into nursing for pre-med."
Terrific.
Okay, Eleanor, you're up.
All right. My name is Eleanor April, and I live in right outside of Philadelphia, Pennsylvania, and I go to Germantown Friends School. I'm a junior this year. I know I want to go to college. I have no idea where, and I'm not sure what I want to major in. I think something in the sciences.
Oh.
I'm considering engineering or pre-med.
Terrific. Terrific. So seems like only yesterday I was in your position of trying to decide what I wanted to do, and but I was always interested in biology, so it wasn't a very difficult choice for me to major in biology, and I went to UC Davis studying biochemistry, and as an undergraduate there, I did a internship. And the one thing I want to say to all of you right now is, no matter what you're thinking of doing in college, get some experience. Do a lab rotation or do rotations with physicians, nursing, whatever you're interested in. Find out if that's what I did, and in fact, that's how I got my first job. So because I had laboratory experience, I didn't know what I wanted to do after I got to college, whether I wanted to go to the center, whether I wanted to become a researcher.
Sorry, Dan. Eleanor, can I just ask you, if you are typing, sorry, Dan,
Does that make it difficult to hear?
A little bit. What you have to do is just mute your microphone, and then you can type to your heart's content. I don't think we'll hear anything. Okay, and then when you're ready to say something, you can unmute yourself.
Are you multitasking, Eleanor?
Oh no, I was taking notes actually.
I could, yes.
I'm not sure what we have to
You're
write
fine.
about this session, so I thought I'd just record everything that's going on. Yeah.
So, as I was saying, because of not knowing what I wanted to do, I had this internship, and I was able, fortunately, a little bit of luck always helps. I was able to get a position in a pharmaceutical company called Genentech. Are any of you familiar with that company? Or heard about that company before? Well, back in my day, when some Genentech was the biotechnology equivalent of Apple Computer, which you probably know about, or Bill Gates and Microsoft. And it was this brand new type of company that could promise to change the pharmaceutical world, in which it would actually make human proteins in bacteria, and human proteins that could be used as therapeutic reagents. And it was this serendipity of me wondering what I was going to do and finding this terrific job at Genentech, and found a very exciting place to work where we can all pull together as a team and try and build new medicines. And so that kind of got me hooked on the pharmaceutical research area. And after several years at Genentech, where I worked on a variety of projects, and we can maybe talk about some of those later, I decided to go and get a PhD doing research. And to do that, I went to UC San Diego, and I worked in a laboratory where we were trying to identify the proteins responsible for developing the pituitary gland. And the pituitary gland is a gland that makes all kinds of hormones in the body, including human growth hormone. It makes thyroid hormone, and thyroid hormone is one of the hormones that sets your metabolism. So if you're depleted in thyroid hormone, you'll gain weight, you'll be lethargic, you'll be depressed, and there are a lot of, perhaps you even know some people who take thyroid supplements. It's quite common. This is quite a common problem. So in that research, I found some of the proteins that were responsible for that. And then because I loved this pharmaceutical research area so much, when I graduated with my doctorate from UC San Diego, I got right back into the pharmaceutical world, where if you've read my bio, you see that I've worked on all kinds of different medicines. It's very exciting research. It's very different probably from all the positions you're talking about, so I kind of expect lots of questions about the field. And currently, I'm working on medications for the eye. This is a very exciting area for me. I personally love that area of research, and been working on it in that area on and off for quite a while. And so I'm working now on drugs for the treatment of macular degeneration. Anybody hear of that or know somebody with macular degeneration? No?
No.
It's quite common.
We had a
I've heard of it. Yeah.
we had a student yesterday who on the call with Dr. Drolet, whose grandmother has the disease. But go ahead, Dr. Drolet.
Well, it's the leading cause of blindness in people over 60 years old. And it's called age-related macular degeneration because it's a disease of the elderly. It generally happens to people over 60 years of age, and more in women than in men. And so what happens with this disease is there's the retinal layer in your eye, which receives the light and it transmits that light information to your brain. And then there's another layer of cells underneath the retina, which nourish the retina, keep it alive. And those cells start to die. And when they die, then the retina above those cells die. And there you get holes in your vision. And it becomes really catastrophic when it becomes an advanced form, where you get blood vessels that grow into the retina and they leak, hemorrhage, and then destroy your retinal layers. So in the last 10 years, we've been able to make, the pharmaceutical industry has made quite a bit of progress in developing treatments for this condition to extend eyesight now for several years at least. And so that's what we're trying to do. One of the new thinking in the area is that it's our own body that begins to start attacking these cells. It's what we call the innate immune system. It's not actually the antibody, but it's this immune system that everybody has when they're born called the complement system. And the complement system can kill foreign cells and bacteria and viruses without the help of antibodies. But it's thought that as we get older, this system starts to attack our own cells and therefore causing damage. And so if we want to be able to slow down this complement system, and so here at Somologic, we're working on inhibitors of this system for age-related macular degeneration. That's currently what I'm working on now. So that's kind of a brief run-through, which you probably read in the bio already. So I'm really anxious to hear and answer any questions you guys may have about the field of pharmaceutical research or graduate work or college, what it's like to do work in this in college, or anything you may be interested in.
I had a question actually. What type of AMD are you working on right now?
So that's a great question. There are actually two types of AMD generally. One is called dry form of AMD, and actually everybody first gets that form. So the physician or even the optometrist who looks in your eye is going to be able to diagnose that you have age-related macular degeneration by these little yellow white spots that form on your retina. And you can just see it by looking at your retina. The spots are called drusen, and what they really are believed to be are the waste products that these nurse cells that I spoke of can't efficiently process as they could when they were young cells. And so they kind of just spit them out in these globules, fatty-like and full of denatured proteins. It's full of all kinds of things called drusen. And sometimes then where this drusen can build up, and then the waste product then can actually contribute to the death of these cells. Also what can happen is the waste product can then damage this membrane we call Brooks membrane that's behind these cells. And once that happens, then you'll get the other type of age-related macular degeneration called the wet form. It's called the wet form because blood vessels start growing into your retina that aren't supposed to be there. And these cells leak and they hemorrhage. You can imagine what that's like. It's sort of like a little stroke in your eye. And then it quickly damages the eye. And if you get the wet form of AMD, without treatment, you'll be blind in that eye within 6 months to 2 years. So it's that form of AMD that just in the last 10 years, we have treatments for. Before that, there wasn't anything that was good at all. One interesting thing for treating the dry form of AMD, the only treatment for that now are these eye vitamins. So you hear a lot of vitamin studies. Most of them prove not to be effective. But in this case, vitamins for the eye, once you have the dry form of macular degeneration, vitamins actually can slow down the process of the development of it. So we're working on both kinds of it. We think complement inhibition would be important for the dry form as well as the wet form.
I have a question.
Sure.
You mentioned earlier that older women are more likely to have AMD than men. Could you explain why?
I don't know. Maybe you can solve that one. It's just, and it's not because women live longer. That's true. They do live longer. Mm-hmm. But if you take the age component out of it, women are more likely to get this disease, and it's not clear why.
Okay.
What's your favorite part about working in the field that you're working in?
Well, my favorite part, the favorite part, that's a pretty easy question, actually. When you develop a medicine that actually helps someone, or helps a group of people, that's a tremendous thrill. To be part of a team, come up with something that makes somebody's quality of life better or extends their life, so they have more time on this earth. That's a great feeling. That's the best part of the job. And I have to admit that only about 1 out of 10 of the projects, an average pharmaceutical scientist, 1 out of 10 or 15 of his projects actually goes on to approval. So it's a game of a lot of failures, but the one or two successes really make up for that.
Dan, Dakota is texting. She wanted to ask you when you started to become interested in the eye. Was it something you were always interested in, going way back, or what sparked that?
Well, to be honest, it wasn't something I had thought about until I was asked to join the project team for the very first time. This would have been in the early 2000s. I was asked to join a team to develop what became Macugen, a very successful pharmaceutical drug. The first what's known as an anti-vascular endothelial growth factor treatment for wet AMD. I'll explain that in a minute. And so it was the opportunity to join a project that I found that I was really fascinated with this area of medicine. And so it was fate or luck. And then I've always tried to stay in that field once I fell in love with it. So it was just an opportunity, and finding out I liked it. But let me explain what I just said. Vascular endothelial growth factor. It's a protein. So I mentioned that the devastating wet form of AMD is this growth of blood vessels that grow inappropriately into the eye. And one of the protein, it's like a hormone, and what it does is it tells the blood vessels to grow. Vascular blood vessel, endothelial, that's the type of cell what vessels are made of, and growth factor, just that. It tells the cells to grow. So if you stop the function of vascular endothelial growth factor, you'll stop the growth of cells, and was proven to be the case, and it worked in this condition. It's also is used, as you might imagine, different vascular endothelial growth factor inhibitors. They're used in cancer. Obviously, cancer, to grow, cancer needs to recruit new blood vessels in order to grow. And so if you can stop the growth of those new blood vessels, you can slow down the cancer, and that's proven to be true as well. So it's been a very useful class of inhibitors pharmaceutical world, and they've all been developed in the last 10 years.
And so, Dan, let me just clarify. Are you saying then that the first drug ever for this kind of disease was one that you worked on?
The first one that was that for ocular indication was the one I worked on. It didn't turn out to be the best one, guys, but it was the first one. Yeah, it's a competitive field. Other drug companies came up with even better ones. And so now it's our turn to make an even better one. So that's how it goes. Patient wins in that case.
All right. Carter, how about you? Do you have a question? Oh, by the way, Carter joined the call late. Carter, maybe you could tell us a little, just quickly introduce yourself and tell us what you might plan to study in college.
Okay. Well, I'm Carter. I go to the school that Mr. O'Connell teaches at St. Andrew's School. In college, I actually am not very interested in studying medicine so much as maybe science and international.
I think that was
that answers the question.
You were a little bit broken up there, Carter.
It's this bad Wi-Fi.
All right, it looks like the message got across.
About that.
Yeah, no, and I am interested in science. I've kind of recently thought about other other different career paths, and I do want to go to liberal arts college, at least at first. I'm not really quite sure. There's like a whole world of things I want to do, and I can't do all of them, but I'm working on it.
Yeah. I understand completely. I have two daughters, and 16 and 14, and they're starting to have to make these decisions as well. So it's a tough time. Yeah.
Probably oncology would be the study of medicine that I'd be most interested
Wow.
in. Yeah.
We need more of those.
Yeah, both my aunts had breast cancer, so I feel kind of motivated to, want to work with that.
Interesting, yeah. I hope they're doing well.
They are. They're actually both in remission. So
Great.
My other question, I may have missed this earlier, but did you always want to do research as your sort of career, or what was your original, I guess, intention or career path when you wanted to be, when you were thinking about going to medicine and science?
Yeah, I think, as I said, I thought about being a physician quite a bit, but I also thought about doing fundamental research. But what did it for me was actually the internship that I did in college. I really enjoy working in the lab. It makes me weird, I guess, but I found that fun. I liked working with my hands. I liked the aspect of doing an experiment and seeing what the result was, and it would open new questions to ask and answer, and that kind of give and take of generating a hypothesis, doing experiments, and seeing the results from your own hands. That was a lot of fun. I thought that I wouldn't have that as an MD, and probably not as much, but I do want to make clear that you can pursue both. Maybe I wasn't so clear when I was your age or an undergraduate student, that you can get an MD and then you can do research. So they're not mutually exclusive by any means. I probably didn't understand that then, but certainly I'm very happy with the choice I made, because I still get to work in the lab some, and
That's
I still
really cool.
enjoy it.
Yeah, I'll just brag a little for Dan. Dan's a senior director. He works up in the offices, with the other top administrators at SomaLogic, and none of them go in the lab. But Dan also finds the time. He gets in early. He leaves late. He finds the time to come right in and right next to the the slobs like me in the lab, and it's such a boost to everyone's morale when the upper ups.
Right. I was just going to say the opposite, because then they have the boss standing over their shoulder looking at their experiments too. I don't know if that's all
shaking as they're trying to do it.
No, not at all. Not at all.
But they can't have all the fun. They just can't.
No.
Dakota had another question. She asked, what are some of the most interesting topics that SomaLogic is researching currently?
Yeah, I think that's a great question. I probably should explain a little bit about the technology first. That SomaLogic, even though we are looking at some therapeutic applications, SomaLogic is a biomarker company, really. It's looking at proteins as signals for disease. So you're all pretty familiar with human genetics, right? Looking at the human genome, looking at genes to predict whether you may get breast cancer, the BRCA genes, or whether you may have genes that make you more susceptible to diabetes or what have you. And that's all well and good. But often they can't tell you when you're going to get a disease. So you may have genes that say, oh, you have a very high likelihood of getting diabetes or breast cancer or colon cancer, but they can't tell you when. So how do you find out when someone is going to get a disease? And we believe that the proteins in your body can signal that. So what we've done is made these things we call somomers. They're these reagents that actually specifically bind to a protein in your blood. So you can imagine the Human Genome Project has said that we have a little over 20,000 genes that code for a protein. But actually some of those proteins through alternative splicing get rearranged, and we have even more functionally different proteins than the 20,000. So if we can measure in your blood how much of these proteins there are and watch them change over time, and that we may be able to use those to say, aha, now we're starting to see a very early signal that you might have the first start of breast cancer. We better intervene now. Your gene said that that was coming, possibly, and now we say it's now. That's our goal. We're a little bit away from that, but we're moving in that direction. One of the things that we have done recently is what we call a cardiac panel. So we measure a little over just 1,000 proteins at a time in a patient's blood. And we've looked at patients who's had a cardiac event. Maybe they've had a heart attack, or maybe a valve problem or something like that. And we want to know, once they've received treatment, who's at risk for another cardiac event. So here's a problem where someone's had a heart attack and they've survived it, doing okay. How do we assess how likely they are to have another heart attack? And we've been able to apply our technology to actually stratify these patients into those that are extremely high risk for another cardiac event, from those who are maybe at medium risk to those at low risk. And now a doctor would have something definite to say, okay, this person's looking good now, but maybe they're very high risk for another event. Maybe we ought to be a little more aggressive in our treatment of this patient, where this other patient population, they're doing fine. They don't need so much care. So that's one thing that we've recently done, I think is pretty exciting. Another thing that we've done has been a collaboration with this group at Harvard Medical School, where they did this tremendous technique called parabiosis. So for over 150 years, this has been done. And what you do is you actually take two mice and you put them side by side and you tie their circulatory system together. So the blood is shared between the two mice. Isn't that kind of wild? And so it's been known for a long time that if you take an old mouse and you put them next to a young mouse and tie them together. Now, the old mouse, we know, I should put it up here as a thing. The old mouse has cardiac problems, so they have hypertrophy. Are you familiar with that term?
We just to remind you guys, Tui, the last case we had, the cardiology case, he had an enlarged heart. He had cardiomegaly, and
okay.
Sorry.
So hypertrophy just means that the cells are big. So the cells in your heart become big and floppy with age. I'm getting there. So and then you also get thickening of the heart, and then the electrical impulses around the heart don't work as well, and the heart doesn't pump as efficiently. It's called diastolic heart. It's very common in the aged. And it's been known that if you do parabiosis and put a young mouse next to an old mouse, that something in the blood would transfer to the old mouse and reverse the thickening and the hypertrophy of the heart. So, but for decades, nobody could find out what that thing was. What is that? And people have been looking for a long time through all kinds of different techniques, looking for metabolites, looking at the genes, looking at the RNA. And so finally, this group at Harvard came to us, SomaLogic, and asked us to apply our technology to look at these blood proteins, these over 1,000 different blood proteins, and see if we could find differences between the old mouse and the young mouse that could possibly explain the differences that we saw. And so that was done. And what they found a few things that were different. One thing stood out, and that was this thing called growth differentiation factor number 11. We call it EF11. It's actually another one of those growth factors that I talked about. And it was higher, much higher in the younger animals. So that was known to stimulate health. So it's the sun. And how do you think they know that information? What would experiment? So the mouse has high ratio GDF11 is known to work on the heart. And
hey, Dan, can you hear me?
The old mouse has little of it. So what would you do to see if it was? Yeah, I can hear you.
Just 1 second, Dan. We've lost Dakota, and you were breaking up for a while there. Can you still hear me?
I can hear you fine.
Okay, here comes Dakota's back. Can you just sort of rewind for about 30 seconds and do that again?
Sure. So I was asking the budding scientist or the budding non-scientist to think about science for a minute. To say, okay, I've just discovered that I put a young mouse in parabiosis with an old mouse, and there's something in the blood that can make the old mouse's heart look young again. And I just said that what we find is GDF11, this protein, is at a greater concentration in a young mouse's blood than an old mouse's blood. How would I prove that it's GDF11 that was the factor in parabiosis? What would you do?
Find a way to put the GDF11 in the old mouse.
Absolutely.
Yes.
Just that simple. Yeah, beautiful. You're scientists. So that's what we did. People at Harvard did. They went out and got GDF11 and said, okay, is that the factor? And they put it in, and guess what? It worked. It worked. And so now it works in mice. That doesn't mean it's going to work in any other species. So that's the next thing maybe someone has to say. Well, it works in a mouse. Does it work in a rat? Work in a monkey? Does it make a monkey's heart younger? And maybe, does it work in a human? Maybe we have a treatment coming up for diastolic heart failure in a human. It will take probably 10 years before we know that answer, by doing all the careful experiments and clinical trials that it will take to find out if GDF11 will work. So that's some of the things that Some Logic has done recently. It's a great place. It's fun. Lots of fun things happening.
In your biology, or sorry, not your biology, your biography, you mentioned the process for getting drugs approved and all the testing that had to go on. Does that always take around 10 years, like it did for this, like it will take about for this heart problem, or does it take longer or shorter usually?
It can take shorter. I think 5 or 6 years is the shortest from drug discovery to approval. There are exceptions always depending on need. Sometimes things can be shortcutted if people are dying for want of any treatment. They can maybe be sped up a little bit, but generally it takes 8 to 12 years to get a drug approved. Part of that is the discovery time. So obviously you just don't walk into work and say, oh, I'm going to come up with a drug that does X. It takes time to find that molecule that has the properties that you want. So it not only has to have the activity you want, whether that is to inhibit something in your body or to activate something in your has to be non-toxic, or at least as non-toxic as you can get it. And then you have to prove to everybody in animal research still that the drug works, but even more importantly, that the drug is fairly safe. So you can't put it into a human being until you prove to the Food and Drug Administration, or the FDA, that your drug is safe. So that's why we do animal testing. Once you have this package that shows it works and is safe in animals, you submit that to the FDA, and they review it, and then they give you an approval if everything's good in order to test it in humans. So the very first study in humans is what we call a phase 1 study, and that is actually at low doses. You're not trying to cure anyone. You're just looking at the safety. So they're called safety studies. Sometimes it's patients, depends on the drug, but often it's volunteers. College students, to make money, often volunteer for such studies because they can be paid to do the phase 1 volunteer. So you do these studies in humans with starting with low amounts of drug, and then you start to scale up. And if your drug is safe, that's what the FDA cares about most, then you apply to the agency for approval to do a phase 2 study. And a phase 2 study is actually what's called an efficacy and dose ranging study. So efficacy means basically, does it work? Is it efficacious? Does the drug do what you want it to do? That you find out in phase 2 studies. You vary the dose again. Now you have some more patients than you did in the phase 1 study, and you began to ask the important questions of how much drug do I give, and does it work? Once you get that information, you put it in a package, and you submit it to the FDA again, and they review it, and they decide whether you can do phase 3 studies. Phase 3 studies are these big clinical trials that maybe you've heard about, which often have several hundred or even several thousand patients. And this time it's a controlled study. So not only do we put in our drug into, say, half the patients, but we could have to put in placebo or something else into the other patients. And so then we monitor the side effects in both groups. And yes, there are side effects in the placebo group. Some people take something and something happens to them. It's a side effect. They might get a headache. And so you have to score that. Sometimes, actually, you give them a placebo and they get efficacy. You just because randomly in their body, they're fighting off the disease or the indication, and so they might get better on their own. And so that's why we have that group. And then we have the treatment group with our drug. And so this is a big study, and then we do statistical analyses, and we ask, in the end, was our drug safe? Always the most important thing. And did it work? Did it work enough better than the placebo to actually help people? If we think so, then we submit that package. Now we are submitting something called a new drug application. It's the government, so it's called an NDA. It's like NASA. Everything's an acronym. So we submit that, and then the agency reviews the package, and they actually get a group of physicians that review the package for them. So an outside council of physicians in that field, and they all review the data and make a recommendation to the FDA. And then the FDA decides whether or not your drug can be approved. And if they do approve it, you're not done yet, because then the FDA may ask you to do more studies. They'll approve it, but they want you to do this study or that study, perhaps in a different group of people, or maybe to look for interactions with other drugs, all kinds of things. So you're always studying your drug. And that's the process. And that's why it can take a while. We try and be very careful. The last thing, we mostly only get press in the pharmaceutical world when we hurt someone. It does happen, but we do go to great lengths to avoid that from happening. And that's the process, and that's why it can take quite some time. And you can imagine if you're running, say, a 3,000 patient study where you're paying for everything, you might want to take some time to consider exactly how you want to do that study before you initiate it. Because if you do a 3,000 patient study and you've asked the wrong questions and measured the wrong endpoints, then you just wasted millions and millions of dollars. So people are very cautious in the human clinical realm. Does that answer the question about the process?
Yeah, it does. I was just curious, how much does it cost usually? Or does it vary a lot? I mean, I'm sure it's a lot.
Yeah, that's a great, there are a lot of estimates.
Terrific question.
It is.
Yeah.
And, first, let me say that about 1 in 10 drugs that go into human clinical trials actually get approved. So even after you've done all that research and start those studies, often the drug doesn't work well enough or at all. And so it's gone, and nobody hears about those failures. So how much a study costs depends on the indication often.
The disease.
I can explain that by if you're doing a cardiac study, that you have to pay for all the cardiac testing, all the lung testing, and those can be very expensive per patient. Whereas, and those studies can go on for months. So the patient enters the study, starts taking your medication, and they might be on study for 6 months to a year. So that takes a long time, and you're paying for many visits to the doctor. But if you're studying, say, an antibacterial, somebody who has strep throat, and that usually only takes 2 weeks, and you don't have to do much testing of them. Their sore throat goes away. It doesn't look red anymore. They don't have a fever. Very cheap endpoints to do. So there's a wide range of maybe, I would say, in the range of $100 million, say, to $1 billion to develop a new drug, depending on the field that it's in and how many patients you need. Another thing that goes into it is how big a study you have to run to prove your medication works. But the details of that are probably excruciatingly boring.
Well, they're actually the details of that are on the front page of today's New York Times. The statin study came out one way with a small sample, and then came out the other way with a large sample.
Yeah.
Just today, very very interesting. But anyway.
Yeah, the danger of a small sample size. Right. If you only do 10 flips of a coin, you'll get 7 or 8 heads, and you might think it's an unfair coin. But if you did 7,000 flips, it'll probably come out pretty close to 50-50.
All right, I think we might have time for one more question, and then I'll have to let Dr. Drolet get back down to the lab.
That's where I will be going.
Here we go. We have one coming in from Dakota. Thank you, Dakota. She asks, is there a lot of statistical analysis involved in your research?
Tremendous amount. Statisticians, I sometimes fear, are taking over the world. It's the era of big data. Have you heard this phrase before? Big data. It's in the science circles, it's the buzzword these days. With the genome, there's so many genes. So how do you analyze it? You get all these data points. With the proteome that we study, all the different proteins, we have all these, measuring 1,000 proteins at a time. Then we measure them at a second time and a third time, and measure them in 1,000 different patients. So we have this tremendous amount of data. And how are we certain that what we're looking at isn't just this coin flip problem where maybe we're being fooled? And so that's where we hire a lot of statisticians to look over not only how we design the studies, but how the studies are interpreted at the end. And this is actually, I think, more true, or at least as much true in the non-biology realm, with all the data that the government's collecting on us right now as we're doing Facebook or doing Skype. There's, people are reading everything everybody does on the computer. Well, that's thousands of data points, and so I think people who want to push advertisements to you, or the NSA who wants to figure out what you're doing, they are using statisticians to help them handle this big data. So I would, if you like math and you like statistics, I think that that's a budding field that will continue to grow. Lots of statisticians. Is anybody taking statistics in high school? All right.
Oh, that's great, Dakota. I think it's the most undertaught math in high school. Everyone takes. Let me ask this question, Dr. Drolet. You work with maybe 50 other scientists or people who might have a use for a high level of math education. Could you contrast the usefulness of calculus and statistics for people in...
I take calculus.
Yeah.
Well, I don't want to badmouth calculus.
Yes, you do.
No, you don't.
Nobody uses it.
Yeah, no, I agree. It doesn't have very many.
I use statistics every day in my work. I use calculus every so often in my work.
Not even.
No, I do use calculus in my work. Not often, but I do. But yeah, statistics. If you are interested in, but I taught statistics to med students for 5 years while I was in graduate school, and I admit I only saw those. So I taught I didn't teach the classes, but I taught the study sessions, and I was the person they came to for help. So I only saw the people struggling with it. But I don't think you can actually be a physician without understanding the rudiments of statistics. It's so important in understanding the results of a clinical trial. So if you're a physician and you want to understand this new study for this medicine, you really do have to have an understanding of what these numbers mean and how they were derived and used, because sometimes they're not used appropriately, even in the scientific literature. There's a recent article in the American Scientist magazine, just this bi-monthly issue, if you want to look at it, which describes the misuse of statistics in regular research papers. So if you're interested in, I don't even think if you're interested in science. I think if you want to be a good citizen, then you should take a statistics class so you understand what's in the newspaper. It's going to affect your health, even if you're interested in Shakespeare for a career.
All right. I think that may be all the time. Yeah, that is all the time we have.
Not too bad. It's fun.
Yeah, thanks so much, Dan. That was great.
Thank you.
All right.
It was fun. Thank you, guys. And good luck to all of you with your college decisions and your careers.
Thank you.
Thanks so much. This was really a really great Skype call. Yeah.
Good.
All right. You all can log out. I'm going to stay on with Dan just for another minute.
Okay.
But Carter, Dakota, and Eleanor, thanks for your call, and we'll see you later.
Thank you for all your time.
You're welcome.
All right, Dan. Wow, terrific. That was great. Thank you.
Yeah, I think it's always better the second time you kind of feel where people are.
Yeah.